So by the time you finish listening to this deep dive, your body will have naturally produced thousands of damaged, dysfunctional cells.
Thousands of them, yeah.
Right. And, just a few years ago, those microscopic errors were basically considered a one-way ticket to physical decline.
Like an inevitable tax.
Exactly. The tax we all pay for getting older. But today, right now in the year 2026, we actually have the molecular tools to hunt those specific cells down.
And clear them right out of your system.
Right out of the system. We are officially living in an era where humanity has, well, we've stopped treating aging as this mysterious, inescapable fog, right?
Yes, we started treating it as an engineering challenge.
A highly complex, but completely solvable engineering challenge.
It's honestly the most significant pivot in the history of human medicine.
Yeah.
I mean, we are looking at a fundamental rewiring of how we perceive biological time.
Okay, let's unpack this, because that is exactly what we're doing today. We've gathered a massive stack of sources for you. We're talking 2026 biotech press releases.
The latest clinical trial data that literally just got unblinded.
Yeah, and comprehensive longevity market analyses. Our mission here is to cut through the intense sci-fi hype you're probably seeing all over social media.
Because there is a lot of hype.
So much hype. We want to zero in on the actual clinical reality of human health span extension. And look, we know you're already familiar with the basics of longevity.
Right. You know the difference between simply extending lifespan, just chronological years lived.
Versus optimizing health span.
Exactly. Years lived with high physical and cognitive function. So today, we're going straight to the bleeding edge.
Which we have to do because the sheer volume of capital and, frankly, computational power pouring into this space right now has accelerated the timeline.
Oh, far beyond what anyone predicted even 5 years ago.
Yeah.
We aren't talking about theoretical models for, like, the year 2050 anymore. We are talking about investigational new drug trials happening in human patients right now.
Today, so pull up a chair. You are the third person at this table, and we have a lot of ground to cover.
A ton of ground. We're going from the economics to epigenetic reprogramming.
AI, drug discovery, all of it. But before we get into the lab breakthroughs, I want to look at the macro picture.
Okay.
Why is the entire global medical infrastructure suddenly pivoting toward longevity science in 2026? Like, what finally broke the old system?
Well, the old system broke because it was built on a mathematical impossibility, frankly.
How do you mean?
In the medical field, we referred to the traditional reactive model of healthcare as the whack-a-mole problem.
The whack-a-mole problem, which is incredibly frustrating for patients, by the way.
Absolutely.
You wait for a crisis to pop up, say an oncology report comes back positive, and you hit it with a mallet. Surgery, chemotherapy, radiation.
You beat the mole down.
You beat it down.
Yeah.
But while you're recovering from that, a cardiovascular issue pops up.
Right.
So you hit that with a stent. Then metabolic dysfunction rears its head. It's basically a perpetual cycle of treating downstream symptoms.
It is. And the data from our market analyses reveals just how futile that game ultimately is. Here's a statistic from the sources that usually shocks people. If we magically cured all forms of cancer tomorrow...
Like eradicated it completely from the human species.
Eradicated completely.
Yeah.
We would only add about 2.5 years to the average human life expectancy.
Wait, I really need to pause on that. 2.5 years?
Yeah, just 2.5.
For curing the most feared disease on the planet, how is the impact that remarkably low?
It's because of comorbidities and the biological interconnectedness of organ failure. You see, by the time a patient reaches the age of 80, over half of that demographic has at least 5 chronic illnesses simultaneously. 5. So if you extract cancer from an 80-year-old's biological equation, you haven't actually done anything to address the underlying neurodegeneration.
All right, or the arterial stiffness.
Exactly. The failing renal function, the systemic inflammation. Curing cancer just leaves a void that cardiovascular disease or dementia will immediately fill.
Because fixing the specific pathology of a tumor doesn't fix the root cause that allowed the tumor to grow in the first place.
No, it doesn't.
Biological aging is basically the water that all these diseases are swimming in.
That's a great way to put it. And this brings us to a conceptual framework that David Sinclair from Harvard Medical School highlighted in our sources.
The information theory of aging.
Right. It completely reframes what is happening inside your cells at this very moment. Imagine your cellular biology is like a CD.
Okay, an audio CD. So the actual genetic code, the ones and zeros, the music on the disc, that's still perfectly intact.
Yes. The genetic sequence itself rarely changes. A skin cell in an 80-year-old has the exact same DNA sequence as it did when that person was 20.
So the problem isn't the music.
The problem isn't the music. The problem is that the laser reading the cellular machinery, can't read the information properly because the surface of the CD has become covered in scratches.
Ah, and those scratches are the epigenetic noise we always hear about.
Yep.
The chemical changes caused by environmental stress, poor sleep, toxins, and just, the thermodynamics of living.
Yes, specifically, we were talking about DNA methylation and chromatin remodeling. As you age, your epigenome loses its precise configuration.
Right.
Chemical tags attach to the wrong parts of the DNA, shutting down youthful genes.
And accidentally turning on genes that promote inflammation or cell death.
Precisely. The skin cell basically forgets how to be a resilient skin cell. It loses its identity.
So the objective of this whole 2026 medical pivot isn't to rewrite the digital music, it's to polish the CD.
Polish the CD.
To clear away those epigenetic scratches so the cell can access its original youthful operating system.
Yes. And the reason this shift is happening so aggressively right now isn't just because the science is finally catching up.
Right, it's the money.
It is the money. It's because of a massive macroeconomic imperative. The financial math detailed in our sources is the real engine driving this forward.
Well, let's follow the money then. Because as much as we love the biology, nothing changes the global standard of care faster than economics.
Sad but true. Our sources point to a staggering economic return on investment here. For every single dollar put into proactive health span expansion and preventative biological maintenance, the global economy sees a 4.4x return.
Wow, a 440% ROI.
Exactly. Realized through reduced hospitalization costs, eliminated long-term care burdens.
And critically improved workforce productivity.
Yes.
Because the workforce aspect is the silent crisis no one was talking about until recently. We are looking at a global demographic cliff.
We really are. Fertility rates have collapsed across the developed world. We are top-heavy.
Yeah, the ratio of retirees dependent on the medical system compared to the number of young, healthy workers funding that system has basically inverted.
It has. And the analyses state our options plainly. We either replace the majority of the human workforce with autonomous robotics.
Which is its own can of worms.
Right. Or we fundamentally alter the trajectory of human aging so people can remain healthy, active, and productive for decades longer.
The numbers in the report are just wild. They estimate that extending the average healthy lifespan by just one single year
1 year
generates an estimated $38 trillion in economic value in the U.S. alone.
It's almost unfathomable.
$38 trillion for one extra year of health span. When you look at those figures, it makes total sense why longevity medicine is no longer a fringe hobby for tech billionaires.
Right. It is being integrated into standard clinical practice. It has birthed an entirely new asset class, actually.
Oh, you mean the bioliquidity thing?
Yeah. The industry is now referring to this as maintaining your bioliquidity.
I want to dig into that term, bioliquidity, because it sounds like we are treating the human body like a diversified financial portfolio.
That is precisely the framework. Just as you manage financial capital to ensure you don't run out of money in retirement, clinics are now helping you manage your biological capital. You insure your home against a fire, right? Now, patients are securing bioinsurance.
Right, so banking their stem cells, mapping their genomes.
And rigorously monitoring their biomarkers.
Yeah.
So they have liquid biological assets to deploy when regenerative procedures are needed later.
You are proactively ensuring you don't become a massive liability to yourself or the healthcare system.
Exactly.
Okay, so the economic incentive is clear, and the biological goal polishing the CD makes sense. But practically speaking, as a patient, how do I actually know if my bioliquidity is stable? Like, if I start a rigorous new longevity protocol today, a new diet, new supplements, sleep optimization, I can't wait 30 years to see if I statistically outlive my peers.
No, you can't.
You can't manage what you can't measure.
Which is why the single most important development in the last few years has been the democratization of biological age testing.
Right.
We have witnessed the death of chronological age as a useful clinical metric, really. It's been replaced by the extreme precision of the epigenetic clock.
Now, I know epigenetic clocks have been around for a while. People were spitting into tubes 10 years ago.
They were.
What has fundamentally changed about the technology we are using now in 2026?
To understand the leap, we have to look at how these clocks are trained to read your biology. As we discussed, your epigenome consists of chemical tags, methyl groups, that sit on top of your DNA.
And a metal group is just a carbon atom bonded to 3 hydrogen atoms, right?
Yes. When these groups attach to specific regions of your DNA called CPG islands, they physically block the cellular machinery from transcribing that gene.
It is the ultimate biological on-off switch.
Exactly. So the clock is going in and mapping exactly where those methyl groups are located across millions of potential sites on my genome.
Okay, so it's a map.
Yes. And the first generation of these clocks, like the original Horvath clock, were trained using machine learning to look at those methylation patterns and simply guess your chronological age.
Which, from a clinical perspective, is basically a party trick.
Pretty much.
I have a driver's license that tells me my chronological age. I don't need my DNA to tell me I'm 45.
Exactly. It proved the underlying chemistry was sound, but it wasn't highly actionable. Then came Generation 2 clocks, like Grimmage.
Right.
They took it a step further. They were trained to predict mortality and disease risk. They could look at your methylation and say, based on these patterns, you have a highly elevated risk of cardiovascular failure in the next 10 years.
Okay, that's certainly more useful, but it still feels like a static photograph.
Yes.
It tells me where I'm standing, but it doesn't tell me how fast I'm running toward the edge of the cliff.
And that is the exact limitation that the Generation 3 clocks have solved. The gold standard highlighted across our 2026 sources is the Dundon pace algorithm.
Let's break down Dundon pace, because if the older clocks were like the odometer on your car telling you the total miles you've put on the biological engine, Dundon pace is the speedometer.
A highly calibrated speedometer.
Yeah.
It does not give you a biological age in years. It measures your current rate of aging. It answers the question, at this exact moment in time, how many biological months is your body aging for every chronological year that passes on the calendar?
Walk us through what a result looks like for a patient.
Well, the test yields a single numerical score. A score of 1.0 means your biological pace perfectly matches the calendar. You age 1 year for every year that passes.
Okay.
But let's say your score comes back at 0.8. That indicates you are currently aging 80% as fast as the average population.
Wow.
For every 12 months that go by, your body only sustains about 9.5 months worth of wear and tear.
And conversely, if my score comes back at 1.25, I am redlining the engine.
You're running hot.
I'm accumulating 1.25 years of damage every 12 months. But what makes this so revolutionary for you, the listener, is the sensitivity of the test.
Its sensitivity is its superpower. Because Dundon pace measures the current pace of biological decline, it responds to short-term interventions.
It reacts fast.
Very fast. Our clinical sources demonstrate that if a patient initiates a significant lifestyle change, say they adopt a strict Mediterranean diet, dramatically improve their deep sleep architecture, or begin a metabolic reset using the latest GLP-1 receptor agonists, they can take another Dundon pace test in just 3 to 6 months and definitively see if their biological speedometer has slowed down.
That tight feedback loop changes human behavior. I mean, in the old days of preventative medicine, you basically ate your broccoli on faith and hoped it paid off 4 decades later.
Right, it was all faith-based medicine.
Now you have empirical, personalized data. You can test a highly specific supplement stack, retest your methylation in 90 days, and know with absolute certainty if it is biologically effective for your unique genetic makeup.
And this data isn't being analyzed in a vacuum either. Longevity clinics are pairing the Dundon pace speedometer with massive multi-omic panels.
Oh, right.
They aren't just looking at epigenetics. They are simultaneously sequencing your gut microbiome, your proteomics, your metabolomics, and taking real-time data from your wearable devices.
Which raises a massive logistical issue. No human doctor, no matter how brilliant, can hold 800 interacting biomarkers in their head and calculate the optimal intervention.
No.
It exceeds the cognitive capacity of a human physician.
Which is why our sources show the complete integration of AI clinical intelligence platforms into standard practice.
Like holistic care.
Exactly. A platform like Holistic Care, which was heavily featured in the clinical rollout data, ingests all those multi-omic data streams.
Okay.
It uses deep learning algorithms to find the hidden correlations. For example, noticing that a specific deficiency in your microbiome is driving a spike in an inflammatory protein.
Which is accelerating your Dundon pace score.
Right. The AI connects the dots that are invisible to the human eye.
And by doing the heavy computational lifting, these AI platforms are saving clinical practitioners, what, an average of 15 hours a week in data analysis?
That's right, 15 hours.
That is nearly 2 full work days handed back to the physician. But does that make the doctor obsolete? If the AI is calculating the biological math, what is the human actually doing?
It elevates the physician's role entirely. They transition from being data analysts to strategic health architects.
Health architects, like that.
Yeah. The AI provides the blueprint of where the biological engine is failing, and the human doctor uses their clinical empathy and experience to design a realistic, sustainable intervention that the patient can actually implement in their daily life.
Okay, so the AI and the doctor are looking at my Dundon Pace score, and it says my engine is running way too fast. I'm at 1.2.
Okay.
The multibromic panel shows a massive spike in systemic inflammation. The speedometer is flashing red. What is the very first biological lever they pull in 2026 to hit the brakes?
The first step is clearing out the microscopic pollution that is actively degrading your healthy tissues.
Yeah.
We have to initiate the war on zombie cells.
Yes. This is one of my absolute favorite topics in modern biology, senolytics. But before we talk about the drugs, we need to understand the enemy. What exactly is a zombie cell?
Well, the clinical term is a senescent cell.
Yeah.
Throughout your life, your cells undergo stress. They are exposed to UV radiation, environmental toxins.
And the simple mechanical errors of DNA replication during cell division.
Exactly. And when a cell accumulates too much structural damage, it is designed to initiate a process called apoptosis.
Programmed cell death.
Programmed cell death. It politely sacrifices itself so the immune system can clear it away, making room for a healthy new cell.
It's biological altruism.
Yes.
But the system isn't perfect.
No, it isn't. A small fraction of these severely damaged cells refuse to die. They evade apoptosis.
So they just stick around?
They stop dividing, they cease performing their designated organ function, and they simply remain anchored in the tissue.
They become cellular squatters, just sitting on the couch inside your liver or your joints.
Yeah.
But if they've stopped dividing, why are they dangerous? Why not just ignore them?
Because they aren't dormant. They are metabolically hyperactive. These senescent cells begin secreting a highly toxic, localized cocktail of inflammatory cytokines, chemokines, and tissue-degrading enzymes.
Gross.
Very. In the longevity field, this is known as the SSP, the senescence-associated secretory phenotype.
The SASP. And those cytokines, things like interleukin-6 and interleukin-8, are basically biological acid.
They are. They actively dissolve the extracellular matrix, which is the structural scaffolding that holds your tissues together.
Wow.
And worse, the SASP is contagious.
Wait, contagious?
Contagious.
Yeah.
Those inflammatory signals can penetrate the membranes of nearby, perfectly healthy cells, corrupting them and forcing them into a state of senescence as well.
It's literally a localized zombie infection.
Literally.
One damaged cell in your knee joint infects the surrounding cartilage cells, and suddenly you have osteoarthritis.
Yep.
If they accumulate in your blood vessels, the vascular walls stiffen and you have hypertension. This chronic, low-grade inflammation driven by the SASP is what researchers call inflammaging.
Inflammaging is recognized as a primary driver of almost every age-related disease. So the clinical objective is clear. We need a mechanism to selectively hunt and destroy these senescent cells without harming the billions of healthy cells surrounding them.
Right. We need senolytics.
Exactly.
Well, let's open up the 2026 pharmacy then. What are we using to clear the debris? Our sources highlight a few different tiers of senolytic compounds, starting with naturally derived molecules.
Yes. The foundational research heavily utilized flavonoids compounds found in plants, quercetin, which is abundant in apples and onions, and fisetin, which is highly concentrated in strawberries.
Both of which have demonstrated potent senolytic activity.
Very potent. In clinical applications, quercetin is frequently paired with the leukemia medication called dositinib.
The D plus Q protocol.
Yep, that's the one. D plus Q was one of the first protocols proven to effectively clear senescent burden in human adipose tissue.
Okay, so we have the natural compounds, which are relatively accessible, but the pharmaceutical industry has developed much heavier artillery.
Oh, absolutely.
Let's talk about the synthetic drugs, specifically navitoclax, also known as ABT-263. How does a drug like that actually force a zombie cell to finally die?
Well, it requires an understanding of how the zombie cell evades death in the first place. Senescent cells survive by upregulating a specific family of proteins called BCL2.
Okay.
These proteins act like a biological shield, blocking the internal signals that are trying to trigger apoptosis.
I see. So what does navitoclax do?
Navitoclax is a targeted inhibitor. It chemically binds to those BCL2 proteins and disables the shield.
Ah.
The moment the shield drops, the accumulated damage inside the cell triggers immediate self-destruction.
It sounds incredibly elegant, but wait, if navitoclax is this potent at clearing out the root cause of inflammaging, why isn't this the most prescribed drug on Earth? Like, why isn't it handed out like aspirin?
Because biology is rarely that simple, unfortunately. The massive hurdle with navitoclax is off-target toxicity.
Toxicity.
The BCL2 survival pathway isn't completely unique to zombie cells. Your healthy blood platelets, which are essential for blood clotting, rely on a nearly identical survival protein called BCL-XL.
Oh, no.
So if you administer a systemic dose of navitoclax to a patient, you successfully kill the senescent cells, but you also obliterate their blood platelets.
You induce thrombocytopenia.
Exactly.
The patient's blood loses the ability to clot. I mean, you can't extend someone's health span if you put them at risk of a catastrophic internal hemorrhage.
Precisely. The toxicity profile meant that systemic daily dosing of powerful senolytics was a dead end.
Right.
The field had to completely rethink drug delivery. We needed molecular sniper rifles, not shotguns.
And the sources detail exactly how they solved this. Let's look at antibody-drug conjugates, or ADCs. This is fascinating engineering.
ADCs are a triumph of targeted pharmacology.
Yeah.
Researchers engineered a synthetic antibody designed to seek out and bind exclusively to a specific protein marker that is only expressed on the outer membrane of senescent cells, such as the B2M protein. Attached to this antibody is a microscopic toxic payload.
So the antibody circulates harmlessly through the bloodstream, completely ignoring the healthy blood platelets.
Right.
It only latches on when it finds the B2M marker on a zombie cell. Once it docks, it injects the toxin, killing the squatter without collateral damage.
It radically alters the safety profile. Additionally, clinical protocols have shifted entirely to hit-and-run dosing.
Explain hit and run, because it goes against how we normally think about taking medication.
Traditionally, you take a blood pressure pill every single day to maintain a steady state in your bloodstream.
Right.
But senolytics don't work like that. Clinics utilize an intermittent schedule. A patient receives a high, targeted dose of senolytics over a period of 2 or 3 days to aggressively clear out the senescent burden. Then, they stop taking the drug entirely for several months.
Because it takes time for new zombie cells to accumulate. You clear the brush, let the healthy tissue regenerate, and then come back a quarter later to do another sweep.
Exactly.
Yeah.
But what if a patient's specific pathology makes inducing cell death too risky, even with targeted delivery?
Are there other options?
The 2026 landscape offers an alternative approach. If we can't safely execute the zombies, we can gag them.
We chemically tape their mouths shut so they stop screaming those inflammatory cytokines into the tissue.
Exactly. This is the mechanism of xenomorphics. Unlike senolytics, xenomorphic drugs do not kill the cell.
Okay.
Instead, they modulate the cell's internal signaling networks. They suppress the SAP, dramatically cooling the localized inflammation while leaving the cell physically intact.
And the undisputed king of xenomorphics right now is rapamycin.
Rapamycin is a macrolide compound with a remarkable history, initially discovered in the soil of Easter Island, actually.
That's right.
It is an FDA-approved immunosuppressant, but in longevity applications, it is used at much lower pulse doses to inhibit a central cellular pathway known as mTOR.
Mechanistic target of rapamycin. It is basically the central nutrient-sensing pathway of the body, right?
It is the biological general contractor. When mTOR is highly active, it tells the cell we have plenty of nutrients, build new proteins, grow, and multiply.
Right.
But when you inhibit mTOR with rapamycin, you trick the cell into thinking resources are scarce. The cell shifts from a state of growth into a state of deep cellular repair and conservation. In senescent cells, down-regulating mTOR effectively shuts down the energy-intensive production of the SOP cytokines.
So for you listening, as you navigate the modern longevity landscape, you must understand this core distinction.
It's vital.
If you are looking at a clinical protocol or a high-end supplement stack, you have to ask the physician, is this intervention senolytic, meaning we are trying to clear the debris and induce cell death? Or is it xenomorphic, meaning we are trying to quiet the inflammatory noise?
They are both vital tools, but they operate on entirely different biological pathways.
Right.
And while clearing the debris and quieting the noise is essential for maintaining health span, it is still fundamentally a maintenance strategy.
Yeah.
It addresses the consequences of cellular damage. But it brings us to the most profound question in modern biology.
Yeah.
What about the healthy cells that are still alive but functioning poorly because their epigenetic CD is heavily scratched?
Right. Maintenance isn't reversal.
Exactly.
Can we actually run the biological code backward? Can we take a living, damaged human cell and hit Command-Z to restore its youthful state?
We are entering the realm of partial cellular reprogramming.
Here we go.
And to understand the gravity of the ER100 clinical trials happening right now in 2026, we have to look back at the Nobel Prize-winning work of Dr. Shinya Yamanaka.
The Yamanaka factors are the absolute bedrock of age reversal technology. Let's unpack exactly what he discovered, because it sounds like magic, but it's just pure, elegant chemistry.
Yamanaka discovered that cell differentiation, the process by which a blank stem cell becomes a specialized heart cell or neuron, is not a one-way street.
Okay.
He identified 4 specific transcription factors: OCT4, SOX2, KLF4, and TNIK. Collectively, they are known as OSKM.
And a transcription factor is basically a protein that binds to your DNA and turns specific genes on or off.
Precisely. Yamanaka proved that if you introduce the OSKM transcription factors into a mature adult cell like a skin fibroblast, those factors forcefully rewrite the epigenome.
They strip away all the accumulated epigenetic marks.
They literally erase the scratches on the CD, reverting that adult cell all the way back to a pluripotent embryonic stem cell.
The cell becomes a complete blank slate again. But here is the massive problem, and I'm sure you listening are already putting this together.
It's a de-differentiation problem.
Right. If I want to rejuvenate my liver, and my liver cells suddenly revert into embryonic stem cells, my liver dissolves.
It loses its function.
A blank stem cell cannot filter toxins. It loses its functional identity.
That was the first major roadblock, the danger of de-differentiation. Erasing the biological age also erased the cell's purpose. But there was an even more lethal second roadblock. That M in the OSKM cocktail stands for TNIK.
Which is a well-known oncogene, a gene that, when overexpressed, aggressively drives cancer.
Yes. In early animal models, when researchers applied the full OSKM cocktail in living tissue, it resulted in teratomas.
Oh, wow.
Horrific, chaotic tumors comprised of mixed, de-differentiated cell types. Applying the Yamanaka factors was like using a sledgehammer to fix a watch.
So the million-dollar question for the last decade has been, how do we take the clock back just far enough to clear the damage without erasing the cell's identity or causing cancer?
How do we turn the sledgehammer into a scalpel?
Right.
The breakthrough detailed in our 2026 sources comes from Life Biosciences, a biotechnology company building upon the foundational research of Dr. David Sinclair. Their solution was remarkably elegant in its simplicity.
What did they do?
They dropped the M.
They just removed the TNIK from the cocktail.
They utilized a proprietary gene therapy that delivers only 3 factors: OXL4, SOX2, and KLF4, OSK.
Wow.
By removing the primary oncogene, they removed the accelerator that was forcefully driving the cell all the way back to a pluripotent state.
This is the definition of partial reprogramming.
Exactly.
When you apply just OSK, the factors go into the nucleus, they repair the chromatin structure, they restore youthful mitochondrial function, and they clear the epigenetic noise. The cell gets younger, but it never forgets its identity. A retinal ganglion cell remains a retinal ganglion cell.
It resets the biological clock to a youthful state while preserving the cellular memory.
Yeah.
But even with a safer OSK cocktail, we face a monumental regulatory and biological challenge.
Right. Safety.
How do you actually test this in a living human being? You cannot simply inject OSK gene therapy into someone's intravenous line and hope it safely rejuvenates their entire body.
The FDA would never allow that. The safety risks are astronomical, which is why the ER100 investigational new drug trial, the first ever human trial for cellular reprogramming in 2026, is focused entirely on the human eye.
The eye is the perfect biological proving ground. It is an enclosed, immune-privileged environment. The blood-retina barrier prevents the gene therapy from circulating systemically through the rest of the body. You can observe the neurological tissue directly, measuring exact functional changes.
Let's talk about the specific disease they are targeting in this trial, because the stakes for the patients are incredibly high. The trial is aimed at age-related optic neuropathies, specifically glaucoma and a condition called NAION.
NAION stands for non-arteritic anterior ischemic optic neuropathy.
That's a mouthful.
It is essentially a stroke of the eye. Blood flow to the optic nerve is temporarily cut off, causing severe, permanent damage to the retinal ganglion cells.
Oh.
Under normal biological conditions, adult central nervous system tissue does not regenerate. Once the optic nerve is severed or severely damaged, the resulting blindness is permanent.
Because the cells are too old and too specialized to heal themselves.
Right.
But the ER100 therapy changes the underlying physics of the cell. Let's walk through exactly how this treatment is administered, because the delivery mechanism, the inducible system, is a masterclass in bioengineering.
It operates much like a remote-controlled safety switch for your DNA. In the clinic, the patient receives a direct intravitreal injection into the eye. A harmless viral vector delivers the OSK genetic payload straight into the damaged retinal ganglion cells.
But the crucial part is nothing happens yet.
Nothing.
The OSK genes are inserted, but they are completely dormant.
They remain transcriptionally silent. The gene therapy utilizes a TED-on inducible promoter system. The OSK genes will not express themselves unless they bind with a highly specific triggering molecule.
Okay.
In this trial, the trigger is doxycycline, a common, widely available oral antibiotic.
This is incredible. So the patient gets the injection, and their vision remains exactly the same.
Right.
Then the doctor hands them a bottle of doxycycline pills.
Exactly.
The moment the patient swallows the pill and the doxycycline enters their bloodstream, it crosses into the eye, binds to the promoter, and physically turns the OSK genes on.
The reprogramming machinery roars to life. The OSK factors begin polishing the epigenetic CD of the damaged optic nerve cells, and the ER100 protocol calls for a sustained 8-week continuous burst.
Wow.
The patient takes the systemic doxycycline every day for 2 months.
Hold on, let me play devil's advocate here. Even without the M, isn't leaving a reprogramming switch flipped on for 8 straight weeks incredibly dangerous?
It's a valid concern.
What if the cells eventually forget their identity and form a mass? Doesn't making a cell continuously younger risk stripping away its accumulated functional memory?
It is the exact safety question the FDA demanded answered. And the preclinical primate data prove that a sustained 8-week continuous expression of OSK is the sweet spot.
The sweet spot.
Yes, it is long enough to aggressively force the damaged neuronal axons to regenerate and reconnect to the brain, actively restoring lost vision. But it is strictly controlled by the doxycycline. The moment the patient stops taking the antibiotic at the end of the 8 weeks, the OSK expression immediately shuts down. The newly rejuvenated cells lock into their healthy state.
No tumors, no dedifferentiation, just a young, highly functional optic nerve. For you listening right now, take a second to truly absorb the magnitude of what we are discussing.
It's revolution.
The concept of literal biological age reversal, taking an old, damaged human cell and running its clock backwards so it can heal a severed nerve, has officially moved out of the petri dish.
It is happening in human clinical trials right now.
Right now. And if they prove it works safely in the eye, the next logical targets are the liver, the heart, the central nervous system.
It is breathtaking. However, we must confront a massive logistical bottleneck.
Okay, what's the bottleneck?
Getting the OSK cocktail perfected and finding the exact right senolytic delivery method took human scientists decades of agonizing trial and error. Human biology is infinitely complex. If we rely on human brains to manually pipette fluids and guess molecular structures, scaling health span globally will take a century.
Which perfectly sets up the final piece of the 2026 puzzle. To solve aging, we needed a faster brain, and we built one. The entire architecture of drug discovery has been taken over by artificial intelligence.
The defining paradigm shift in 2026 biotech is the rise of generative biology.
Generative biology.
Systems like NVIDIA's BioNeMo have moved us away from theoretical computational modeling into autonomous closed-loop manufacturing.
Let's unpack that. In the old days, and by old days I mean 5 years ago, if a pharmaceutical company wanted to find a drug to inhibit a specific aging pathway, human chemists would synthesize thousands of compounds by hand.
Painstakingly.
They would test them in lab mice, wait months for results, fail, and start over. It cost billions of dollars and took 15 years to get a single drug to market. Now, it's like we've upgraded from a horse and buggy to a teleportation device.
Our sources extensively highlight a company called Isomorphic Labs, which spun out of Google's DeepMind. They have developed what they call a hive mind architecture.
Hive mind?
This is a complete integration of sophisticated AI reasoning agents and physical autonomous robotics.
Walk us through what is actually happening inside an isomorphic lab right now.
It is a continuous 247 autonomous loop. The AI analyzes a complex biological target, say a newly discovered epigenetic enzyme linked to cellular senescence.
Okay.
Using graph neural networks, the AI digitally designs a novel molecule to inhibit that enzyme. It then sends the chemical blueprints directly to the autonomous robotic arms in the wet lab.
It just emails the robots.
Basically, the robots physically synthesize the chemical, run the biological assay, and feed the results back into the AI in real time.
And if the molecule fails, which it usually does on the first try, the AI doesn't need to hold a committee meeting.
It instantly ingests the failure data, calculates exactly why the molecule didn't bind correctly to the receptor, adjusts the atomic structure in the digital space, and instructs the robots to synthesize version 2.0 immediately.
That is insane.
It explores the vastness of chemical space, utilizing reinforcement learning, failing and iterating thousands of times faster than any human team could ever coordinate.
It's relentless. And this isn't just theory. This AI-driven approach is actively putting drugs into human trials right now. Let's talk about Insilico medicine and the massive milestone they hit in 2026 with a drug called MEN2501.
Insilico operates a platform they appropriately call Pharmaceutical Superintelligence, or Pharma.ai, and the target of MEN2501 is a profound biological mechanism called chromosomal instability, or CIN.
Let's explain chromosomal instability because it is a massive driver of the aging process. When our cells divide, they have to copy our DNA and split the chromosomes perfectly down the middle.
And the cellular machinery responsible for pulling those chromosomes apart is incredibly delicate. As we age, the machinery degrades.
It gets sloppy.
Very sloppy. During cell division, chromosomes get pulled unequally. One daughter cell ends up with too many genes, the other ends up with too few. This chaotic genetic drift is a primary catalyst for aggressive cancers and systemic tissue failure.
It's biological static. And what's fascinating is that the AI didn't just find a drug. The AI analyzed massive data sets, identified chromosomal instability as a high-leverage target, and then designed MEN2501 specifically to stabilize the kinetochore,
The physical structure that pulls the chromosomes apart.
the computer basically engineered a microscopic clamp to hold the DNA together during division.
It is bespoke molecular engineering, but here is where we must apply rigorous critical thinking to the generative biology narrative.
Okay, let's look at the blind spots. I'll throw a massive flag on the play.
Go ahead.
A computer program, no matter how many neural networks it has, is operating in a digital vacuum. It doesn't truly know how a newly invented molecule will behave inside a living, breathing human body with a complex microbiome, interacting liver enzymes, and a unique genetic makeup.
That's a very fair point.
Just because an AI designs a drug that docks perfectly with a protein on a computer screen doesn't mean that drug won't trigger massive renal failure when a real patient swallows it.
That is the exact reason why, historically, over 90% of drugs that work perfectly in animal models fail catastrophically in human clinical trials. Unforeseen systemic toxicity.
Exactly.
However, the 2026 solution to that precise problem is the maturation of whole-body digital twins.
Whole-body digital twins. It sounds like something out of a cyberpunk novel, but it is solving the biggest bottleneck in medicine. How does a digital twin actually work?
It is the ultimate realization of systems biology. Instead of testing an AI-designed drug on a single, isolated biological target, biotech firms are utilizing massive computational power to build highly complex mathematical models of the entire human organism.
So before a single drop of this new MEN2501 drug ever touches the tongue of a human volunteer in a phase 1 trial, the AI runs it through the digital twin.
It uses differential equations to simulate the entire pharmacokinetic cascade. The AI models how the drug passes through the acidic environment of the stomach lining.
Wow.
It calculates how these specific CYP450 enzymes in the liver will metabolize the molecule. It predicts whether those newly created metabolites will cross the blood-brain barrier and bind to off-target receptors in the central nervous system.
That's incredibly thorough.
It simulates the cascading effects across the cardiovascular and renal systems simultaneously.
It's essentially a high-fidelity flight simulator for human biology. You crash the digital airplane 10,000 times in the simulation, so you guarantee you don't crash the real airplane with the patient inside.
Exactly. It identifies systemic toxicity and negative metabolic interactions in silico before you ever initiate an in vivo human trial.
Right.
And the implication for the listener is profound. By combining generative chemical design, autonomous robotics, and whole-body digital twins, the timeline for medical breakthroughs has been permanently compressed.
You will not have to wait 20 years for the next revolutionary longevity drug to navigate the traditional pipeline. AI is shrinking the development cycle from decades to mere months.
We are officially riding an exponential curve. Every single technology we've discussed today is feeding into and accelerating the others.
We have massive AI supercomputers designing targeted senolytics and OSK gene therapies, guided by real-time, Dindon-pace biomarker feedback.
All funded by a $38 trillion global economic imperative to preserve human health span.
The convergence is unprecedented in human history. We are watching the complete dismantling of the reactive whack-a-mole medical paradigm.
It's thrilling but also sobering.
Right, because as we pull all these threads together, we have to look at the reality of how this actually rolls out into society. Our sources highlight a sobering economic reflection that we really have to address.
They do.
As this 2026 clinical reality solidifies, what happens to the people who can't afford the bioliquidity portfolio?
The great risk outlined in the market analyses is the rapid emergence of a longevity divide.
A divide.
We are staring down the barrel of a bifurcated two-tier health system. On one side, you have the haves, individuals with the resources to access concierge longevity clinics.
Right.
They receive continuous multi-omic monitoring, custom AI-designed interventions, and whole-body digital twin simulations. They have premium bioinsurance to bank their stem cells and access to episodic cellular reprogramming.
They basically get the architecture for a highly functional, vibrant 120 or 150-year health span.
Yes. And on the other side of the divide, the have-nots remain entirely trapped in the collapsing reactive legacy system.
Back to whack-a-mole.
Exactly. They are relegated to crowded emergency rooms, managing the cascading symptoms of chronic disease with outdated protocols, and burdened by catastrophic out-of-pocket costs as their biological capital depreciates.
If the market forces are left unchecked, the ultimate luxury good of the 21st century won't be a sports car or real estate. It will literally just be time.
Time and the physical vitality to experience it.
That is a remarkably heavy thought. And it is exactly what we want to leave you, the listener, to mull over as you go about your day. The science to halt and reverse human aging is no longer theoretical. The engineering challenges are being solved right now in 2026.
The biological speedometers work. The molecular sniper rifles are loaded.
But as decades of additional healthy life become biologically possible, how do we ensure our economies, our social structures, and our global access models are actually ready for a world where getting older is an option rather than an inevitability?
It is the defining biological and economic question of our era.
It truly is. Thank you for pulling